Hello everyone, my name is Colby Behrens, and today I'm going to talk to you about neurogenomic signatures of parental care in three-spined stickleback. So briefly, parental care is independently evolved in multiple different lineages, and it encompasses a wide array of behaviors, as you can see in the examples on the right. And the complexity and type of care can vary quite a bit across species, populations, and individuals. And it's this variation that makes a trait particularly interesting to try to understand its underlying genetic mechanisms. And these mechanisms have been explored in the past by many different studies. An example on the left by Dulac showed different brain areas that are associated with parental care. And this Bendesky study on the right used a QTL mapping approach to associate loci across the genome with differences in parental care. Unfortunately, many studies use either highly inbred strains or distantly related species to try and understand these mechanisms, rather than using natural variation within a single species. And this is where three-spined stickleback are particularly useful because of their many different populations throughout the world. In particular, there are two populations or ecotypes in Nova Scotia that differ extensively in the type and amount of care that they provide. And I'll refer to them here as just caring and non-caring stickleback. The caring stickleback on the left will fan at his nest and provide just lots of care to his offspring. So here's an example where he's fanning it with his pectoral fin to provide aeration to the eggs. He'll poke at them to remove fungus or any dead eggs. And then he'll also try to protect the nest as best he can from any predators. In contrast to that, we have the non-caring stickleback on the right, where instead of caring for the eggs, immediately after fertilization, he'll swim into his nest, grab eggs with his mouth, and just start dispersing them across the landscape. So if you look at that white circle, you'll be able to see it best. And this male is going to swim in, grab a bunch of eggs, and then just start spitting them out. And then he'll go back to the nest and keep dispersing them until all or most of the eggs have been removed from the nest. So just in these two examples, you can see very clearly that these two ecotypes differ quite a bit in the amount and type of care that they provide. To take a step back though, stickleback parental care is paternal only, so it's only the male that provides the care. And when he enters the breeding season, he'll become territorial, he'll build a nest out of algae, that's that example on the right, and he'll start courting females. And if the females are interested, they'll drop their eggs, and then he'll start tending them for about a week until they hatch. And then even after they hatch, he'll take care of the fry until they disperse. Once they finally leave, he'll start the cycle over again and start nesting and courting females again. But as I mentioned before, we're not interested in just this caring ecotype, we're also interested in these non-caring fish. So this is where the timeline changes a little bit. These non-caring fish won't take care of the eggs for a week or two. They'll remove them and just immediately abandon them. And we know that paternal care is highly heritable in this system. And we know that genes associated with parental care in mammalian species are also differentially expressed in male stickleback once they become fathers. And much of this work was done by a member of our lab, Abbas Bukhari, who took the same species, male three-spined stickleback, and sampled them at different time points across the breeding season. So when they're non-breeding, before they're caring, during care, and then after care. And he took their brains and sequenced the telencephalon and diencephalon regions and used RNA-seq, which is just an unbiased way to sample the entire transcriptome. And he found that there were a number of differentially expressed genes that were unique to these stages, and then also some that were carried over across stages, which really just highlights the fact that these brains during the breeding season are extremely dynamic in their gene expression. So there's a lot changing just across these stages within the fish. So to expand on this a little bit, I collected brains from multiple stages across the reproductive cycle in caring fish and non-caring fish. I collected them during the territorial stage, during the nesting stage, during post-spawn, 30 minutes after fertilization, where these non-caring fish would have just finished removing eggs from their nest, and the caring fish should be fanning and poking their eggs diligently. Then also an egg stage, which is 4 days after fertilization, where these non-caring fish have long since abandoned their eggs, and the caring fish should be deep into their care at this point. And I too sequenced the diencephalon and telencephalon regions of the brain. The telencephalon includes regions like the hypothalamus, diencephalon includes regions like the hippocampus and amygdala, and I sequenced them as well with RNA-seq. And we have a couple of questions about this data set. So we're very early on into the data analysis, so there's only preliminary results here. But just a couple of questions. Do the ecotypes differ in their brain expression? How does brain expression vary across stages? And then how do ecotypes differ as they progress through these stages of care? So to address question one, how do the ecotypes differ in brain expression? This is an MDS plot of all of the samples where each different point is a different sample. The red points are from caring fish, the gray points are from non-caring fish, and then the different shapes represent samples taken at different time points. And you can see from this that a lot of the variation is explained just by the ecotype. So ecotypes seem to significantly differ in their brain expression. For question 2, how does brain expression vary across stages? Again, if I can draw your attention to the top right that shows the different stages, we use territorial, nesting, post-spawn, and egg. And in these examples, everything is using territorial as the control. So nesting compared to control, post-spawn compared to control, and egg stage compared to control. So this is where we'll get the differentially expressed genes from those contrasts. And if the bars are red, that means they were upregulated, and if they're blue, that means they were downregulated. And this is again split between caring and non-caring, and just showing caring now, and we'll have the diencephalon and telencephalon with D and T. So within the caring fish, you can see that there's quite a large transcriptomic response where lots of genes are turning on or off across these stages and across the brain regions. And similarly, you can see that in the non-caring fish, there's also quite a few genes going up or down across these different stages. So there is definitely a large transcriptomic response as they go across these stages. Their brains are highly dynamic as they're parenting and progressing. And of particular interest right now, I'm looking into this egg stage where you can see there's lots of genes that are turning on and off in these caring fish. Not quite as many in these non-caring fish, but still this is an interesting stage to compare because these are where the caring fish would be providing lots of care. Non-caring fish have essentially abandoned all of their eggs at this point. And if we look into that a little bit more to try to address question 3 just a little bit, I'll show you caring on the left, non-caring on the right. And these are all of those DEGs that we had from the previous figure. So 637 unique to the caring, 260 unique to the non-caring, and there's only an overlap of 37 from those DEGs. So within the egg stage, there's very few differentially expressed genes that are shared between the ecotypes. So they do seem to be changing quite a bit between ecotypes as they change throughout stages of care. Obviously, a lot more work needs to be done on this, but we're seeing some interesting patterns already. So just to go back to those questions, do the ecotypes differ? Yes, they seem to. Does the brain expression vary across these stages? Yes, it seems to. And we are seeing some interesting differences across ecotypes, across stages. So already the patterns are looking very interesting. A couple of future directions. I'll obviously start doing functional enrichment as soon as I can on this data set. We have a similar data set from F1 hybrids where I can look at allele-specific expression. And I'm also doing QTL mapping on F2 hybrids, where this is taking advantage of the genetic differentiation between the populations. And hopefully we can map some of these different regions to differences in parental care, and ideally find DEGs that are also going to the same location as these QTL. So with that, I'd like to acknowledge all the help from the Bell Lab and University of Illinois, and I'm happy to take questions.